Hello everyone, my name is Mehran Mansouri from Abiyankar Lab. The title of my presentation is Do Cells Remember Their Training? Exploring topographical memory in cell migration. Imagine that you are in a forest. Depending on the surface of the area, your body adjusts the way it moves, right? You don't move and walk the same way that you walk in a flat area. But how about after leaving the forest? You are still move and walk the way you did in the forest. So, in our body, cells also move through different landscapes, and we call that cell migration. Not only cells can move, but also they can sense the environment and surprisingly remember it. For instance, studies have shown that cells can remember the stiffness of the substrate that they have been previously cultured on. So here we are seeing stem cells. The difference between these two images is that these cells were cultured on a substrate with a different stiffness. These ones were cultured on a soft substrate and this one on a stiff substrate. And just that, in the post-culture, these cells have differentiated into fat cells, while these cells have differentiated into bone cells. In plain words, they have remembered the stiffness of the past, and based on that, their differentiation has changed. So in my project, we are asking a kind of a similar question, but that is about topographical features. Do cells remember the topography of the substrate, the roughness, the aligned ridges, etc.? So here in the first experiment, what I did, I cultured cells on two different substrates. The first one, a flat surface, and second one, a substrate with topographical features that are 1 micron ridges in the vertical directions. And we saw that cells on flat surface, they are randomly oriented and randomly migrating. But on ridges with topographies, they are elongated in the vertical direction and they migrate directionally. For the next step, I want to explore the question of whether cells have topographical memory. What I will do is that I will culture cells on two different substrates, a flat one and ridges one. And then I will detach them and seed them onto a new substrate. If these cells migrate and behave differently, it means that past topographical features have affected them, and basically they remember that. Why this matters? If we discover that cells have topographical memory, it can help us to effectively design biomaterials and tissue engineering approach to obtain desired results in future. Thank you.